very happy to introduce you now to another incredible scientist, Dr. Helen Wilsey.
I'm Helen Wilsey. I'm an assistant professor at UCSF and an investigator with the Chan Zuckerberg BioHub. My lab studies over 100 genes that are associated with autism and epilepsy, one of which is SYNGAP1. And my lab actually uses frogs to do all of this. So in a second, you'll see a picture, there we go, of what this looks like. And so because we want to study 100 genes at once, we need a model organism that's very high throughput and amenable to studying dozens of genes in parallel. But the story I'm going to tell you today is about SYNGAP and a function that's beyond the synapse. So first, let me give you just an introduction to frogs. So the species I use is Xenopus tropicalis. This is a diploid species of Xenopus, and so unlike zebrafish, which have a duplicated genome, Xenopus tropicalis is a very simple diploid genome that's much more orthologous to the human genome, so well conserved at the DNA level. One pair of frogs will lay over 4,000 embryos in a day, so we have a lot of animals to work with on any given afternoon. They develop very rapidly and so that big dish there of embryos on the bottom left, that's a 15-centimeter dish. That's not a Petri dish. So that's how many animals we have. And they develop very rapidly. In under a week, you have a free-swimming, behaving tadpole with complex behavior and can learn. They're cost-effective. My animal care costs are orders of magnitude smaller than rodents. They're great for drug screening. You can just put the drugs in the water with the tadpoles, and they will go straight into the brain. But if you remember one thing about frogs, let it be this. We can make half-mutant tadpoles. So what we do is we catch those embryos right at the 2-cell stage of life, right after that first cell division after fertilization, and we can micro-inject CRISPR-Cas9 reagents into just one of those 2 cells, effectively making a tadpole 5 days later where only the right half of the animal carries our mutation of interest, and then you can compare what happens on that right half of the animal to the contralateral control side to understand what is the function of that gene. Like I said, it's fast, it's 5 days. We can make a guide RNA in 2 days. So you give me a gene, we can knock it out and look and see what it looks like in the brain in a vertebrate in under a week and for $25. So it's high throughput, it's scalable, it's cheap. And this is a typical afternoon in the lab. So neurula stage embryos where we can make thousands of half-mutant tadpoles and then take a look and see what happened. This is our first proof of principle that we did back when I was a postdoc, where we can mutate a gene required for eye pigmentation on just the right half. And so here you can see on the right half of this tadpole has an albino eye, it's lost pigmentation. You can compare that to the contralateral control side that still has pigment, and you can infer that the function of that gene is an eye pigmentation. Okay, so we did this for quite a few genes during my postdoc, and one of them was SYNGAP. And so when we did that, I thought there would be no obvious phenotype, and then we would do a lot of electrophysiology and be thinking about brain activity. But we had the surprising result that in these animals, on the injected side, which in this animal is the right half, their telencephalon region, or their forebrain region of their brain, was very small. It seemed to be specific to the forebrain. The midbrain and hindbrain looked okay. And so you can see in that inset there, that is the forebrain region of the frog. And on the edited side, you can see a smaller brain compared to the contralateral control. So this was surprising, since the historical and beautiful function that's been described for SYNGAP1 is at the synapse. So you would imagine that maybe you wouldn't see these structural changes early on, and this implies that SYNGAP is required for the process of neurogenesis and actually building the cells to make the brain. Now, this isn't a weird frog thing, and you'll see, I think, later today, Marcella's talk. She has a beautiful paper that just came out looking at something similar in human organoids, and she sees the same thing, right? That SYNGAP1 plays a role early on in brain development, far before synapses are even formed. Who does this have to talk to? There we go. So this led us to the key question here is, does SYNGAP1 have a function beyond the synapse? And we were encouraged to pursue this question once we started to look into clinical findings about SYNGAP. And so we looked in the citizen data, so thank you to all the families who participated in generating this data. And as I'm sure everyone is familiar, there's obviously a very high incidence of epilepsy and autism, and you might say, okay, those could both be explained by a synapse-only mechanism. But when we look deeper, we also found quite a few other conditions that I would argue are difficult to explain by a synapse-only mechanism. So 39% of the participating patients had skeletal defects like scoliosis, club foot, things that are difficult to assign to a synapse function. Dysmorphic features, so the face bones are formed from cranial neural crest. Those are not firing neurons with action potentials. Those are migrating neural crest cells that form the face. Respiratory issues, a lot of asthma, and heart defects that are structural. So a lot of heart murmurs, cardiomegaly, and some aortic dissections. So this isn't, I would argue, not consistent with a synapse-only mechanism. Okay, so what else could it be doing? First thing we did was take a look at publicly available RNA-seq single-cell data, and that's been collated by the Human Protein Atlas, and said, well, where is SYNGAP expressed? As you might imagine, all these red bars are different neuronal cell types, so this makes sense. It's certainly at the synapse, and you would imagine it'd be highly expressed in neurons. Also expressed in astrocytes, but there's one more bar there, ciliated cells. Cilia might be a new concept to some people in this audience, and I'll dig deeper into what cilia are and why you should care about them, but this was a very interesting finding that this other kind of cell type, ciliated cells, are coming up here. We looked into a different data set to see if this was robust, and indeed what we found was that SYNGAP1 expression is highly correlated with core cilia markers, FOXJ1, which is a transcription factor that's essential for building the cilium, and RL13B, which is another classic cilia marker. And so what you can see from the single-cell data, and is statistically significant, is that SYNGAP1 expression is correlated with both of these cilia markers. And if we look back at the citizen data, what you'll see is those conditions that are hard to explain by a synapse-only mechanism are classic cilia-related disorders. So for all of these things, it's well known that cilia defects can cause these. All right, what are cilia? All right, cilia come in two flavors. On the left are motile cilia, so this is one cell, and it puts up about 150 cilia, and they actually beat and move. The other flavor of cilia are non-motile or primary cilia. This on the right is a human neural progenitor cell, so you can see that little small magenta thing is the cilium. And all cells in the body, except for red blood cells, I mean, they're weird, they don't even have a nucleus. All cells in the body other than red blood cells have a cilia, including neurons. So all neurons have them too. These motile cilia have a more restricted localization. They're really in the ventricles of the brain to move cerebrospinal fluid. They're also lining the respiratory tract to move mucus. They're also in the reproductive system, both in the fallopian tubes in females and sperm in males. So that sperm tail is a cilium. The other kinds, and all other cells in the body, have a non-motile cilium, also known as the cell's antenna. And so they function in sending and receiving signals. They're kind of like the original synapse. Even algae have a cilium, and for a long time they were thought to be a vestigial structure until it became very clear in the 90s that cilia are incredibly important for organ development, both in the kidney, the heart, and the brain. And developmental signaling pathways often rely on the cilium to signal. So for example, sonic hedgehog signaling cannot happen properly without a functional cilium. And they're also really important for neuronal transmission. So some serotonin receptors only localize to the cilium, dopamine receptors, TRP channels to sense heat and spice, Many GPCRs are specifically localized to the cilium. And here you can see it's very small. And it's usually right close to the nucleus. It's that small magenta thing there. And this is a human iPSC-derived excitatory neuron. And it's well established that cilia defects affect many different organ systems. So this is a review just talking about if there is a cilia defect, all of the different organ systems that can be affected. And you might recognize a lot of these symptoms in Syngap affected people. So ataxia, epilepsy, mental disability, facial anomalies, some congenital heart defects, skeletal defects, hearing loss. So we wanted to pursue this. Does SYNGAP1 have a function at the cilium? As chance would be, Xenopus is a great model for studying cilia. In the sense, we didn't so much expect that Xenopus would turn out this way for us, but cilia are beautiful in Xenopus. This is a 2-day-old Xenopus embryo, 48 hours. It uses eye on the left side, tail on the right. It's an embryo. If you zoom in on those little tufts, they have this regularly spaced, beautiful epithelium with motile cilia. Each one of those little clusters is one cell that puts up 150 cilia that beat to move mucus. And you can see here, here's a movie Alina made of the cilia actually beating. So these kinds of cells are in the ventricles of one's brain and then also in the respiratory tract. To see if this could be a good model for studying the function of Syngap in cilia, we first wanted to establish that Syngap was expressed in the right place at the right time. And just like humans, Syngap in frogs is expressed in tissues beyond the brain. It's also expressed in these cranial neural crest cells. So those are the migrating neural crests that form the facial structures. It's in the heart, it's in the embryonic epidermis, muscles. And if you take a closer look at one of those multiciliated cells in the epidermis, by fluorescent RNA-C2 hybridization, you can see expression in those particular cells. So it's in the right place at the right time to study it. And then Alina, who is here, a graduate student in my lab, took a closer look to see if Syngap was on cilia. And so indeed, but she did this in 2 ways. This is the first one, which is an antibody staining. And so you can see those magenta things are the cilia on the surface of the epidermis. And you see Syngap in puncta along the ciliary axoneme. And then if you go deeper into the cell, right at the surface of the cell, where the base of the cilium is, which are called basal bodies, which are modified centrosomes, you also see Syngap there on the centrosomes. So this is one way it's an antibody. We don't love antibodies. I've been fooled once. And so instead, she also GFP tagged the human version of Syngap1 and put it into the frogs, which is trivial, and said, where does it localize? And indeed, in multiciliated cells, human Syngap1 localizes again to that basal body, the modified centrosome, and also to the axoneme of the cilium, and especially strong right at the surface of the cilium, of the cell, right at the base of the cilium, where there's high levels of polyglutamylated tubulin to stabilize the cilium. With this is now a nice platform to start thinking about patient variants. The first 2 we looked at are here, they're close to the pH domain, and we wanted to ask, do these missense variants change the localization of Syngap1 at cilia? So what I'm showing you so far is what you've already seen is the localization of the reference sequence. And when we introduce this first missense variant, what you see is a mislocalization to the membrane. And so while in the reference case, there's no membrane localization, now you can see this membrane all around the multiciliated cell that's sequestering Syngap away from the cilium into the membrane. And this other missense variant was different. Instead of taking the protein to the membrane, it actually caused aggregation into these big blobs, but again, taking protein away from the cilium and then aggregating. And so we think that this could be a powerful strategy going forward to understanding and dissecting some of the missense variants. Okay, and so then obviously the experiment to do is to try to knock down Syngap1 and see if it affects cilia. We can do our cute trick in Xenopus where one side is our control and the other side of the animal has been mutated. So on the left side here, you can see the uninjected side, 2 different guide RNAs On the left side, they have their beautiful tufts of cilia. On the right side, you get these big bald spots where the gene has been edited, where you actually lose cilia altogether. If we take a zoomed-in look at one of those patches, they look like this. On the left is a control CRISPR where we have mutated a gene. It's just a gene that's not involved in any relevant process. It's involved in pigmentation. Those cilia look great, 150-ish cilia per cell coming out of the cell. However, when we manipulate SYNGAP1, either by CRISPR or with what we call a morpholino, but these days it's called an antisense oligonucleotide. It's a morpholino. By these two different methods, we see the same thing. Some cells have no cilia at all, but then you can find these cells that are just partially affected, where they're trying to make cilia, but they just can't quite make them. And importantly, and this is preliminary, but we believe in it, is that when Alina adds back that same human plasmid with the GFP-tagged SYNGAP, you see a rescue of this phenotype. So this could also be a powerful platform now for studying variants to see if they can also rescue this phenotype or not and assign functionality to various variants. All right, everything I've shown you so far is frogs. And people always say, is this just frogs? So let's nip that in the bud, okay? So what we also did was, I have a buddy next door, David Kastner, who has rats. And so I got some wild-type rats from him and looked, I told you that in mammalian brains, along the ventricles of the brain, there are these multiciliated cells. And so we wanted to see if we could take a look and see SYNGAP there. And indeed, this is now those multiciliated cells in the ventricle of the brain in a rat. And what you can see is that SYNGAP is on those cilia. What's cool here is it's really kind of on the tips of the cilia, which I think is really fascinating. We can talk more about that if you're interested in that. But we don't think this is a frog thing. We think this is going to translate to mammalian models as well, and we're expanding this work now in the rats, and thanks to support from SRF. Okay, I told you there were two kind of flavors. So far, we've only talked about the motile cilia. But I also want to give some time to non-motile cilia, since those are the ones in neurons. And so we wanted to ask if it was also on those kinds of cilia. And so we looked in two different human cell culture models, HEC293 T cells, which are an embryonic kidney cell line. And here we're overexpressing the SYNGAP1 GFP tag construct. And we see it throughout the cilium in that cell type. And then in RBE1 cells, which are a retinal epithelial cell type from humans, again, we're overexpressing SYNGAP, and we see it, again, at the tip of the cilium, but then also at the base. And this work was done by Alina, who is here, so make sure you grab Alina and chat with her. And this was independently replicated by our colleagues in Mark Van Zascher's lab in another cell type, IMCD3 cells from rodents. All right, so what I've shown you today is that SYNGAP1 patients have cilia-related conditions that are hard to explain by a synapse-only mechanism. SYNGAP1 in humans and frogs is highly expressed in ciliated cells. In a wide variety of cilia, SYNGAP localizes there, and patient-derived variants cause SYNGAP1 to mislocalize at the cilium, and loss of SYNGAP in frogs at least leads to cilia defects. And so we think the take-home point here really is that SYNGAP1 has functions beyond the synapse, one of which is at the cilium. And Alina's got a preprint coming soon, so you can check out all the details once that's online. Okay, my last plug is for any families who are here. We are trying to work on biomarkers for these cilia defects. So if the cilia are not properly functioning in the nose doesn't make the right amount of nitric oxide, a gas. And so James is here. Find James. This is his email address if you're interested in participating in this research. It's a 30-second non-invasive test. You put this just superficially in your nose. It measures the amount of nitric oxide. Who knows if this will work? It's a low-risk, high-reward kind of thing. So if you're interested, we can do patients. There's James. Find James, find him in person or email him. He's got a table out in the lobby. It's a 30-second test. Great. We would love to have people participate. We can test any patients over 2 years old, and we can also test siblings or parents. Okay, great. So with that, first and foremost, I want to thank Syngap families for participating in research and for being here and for being wonderful. I want to thank honorary members of the lab, JR and Mike, whose discussions have been incredibly helpful for thinking about this new function of Syngap. Members of the laboratory, especially Alina, who's leading the Syngap work, she is here, go find her, and James, who's doing the nitric oxide, to see if that could be helpful as well. And I thank our sources of funding and of course, the SRF. So thanks for your attention. If I have time for questions, I'd love to take some. How'd I do on time?
You are on time. You don't have time for questions.
Good.
I just want to emphasize, James is here. He's out here today and tomorrow. Family members, kids. If your kid is here, let's get a reading on them. If their siblings are here, let's get a reading on them. What you just saw is someone go from frogs to potential implications for therapies to here's how we can do it clinically. Absolutely amazing. And we are really excited to support her work.